Hello everyone. Today we are going to study about peptide bond. It is one of the very important bond responsible for primary structure. Suppose imagine these are all amino acids. Like carbohydrates, many sugars join together by glycosidic bond. These amino acids join together by a bond called peptide bond. So these are all amino acids. Maybe imagine this is glycine, this is phenylalanine, this is cysteine. So like this. So these are all amino acids, and these amino acids are joined together by a bond. So this bond between two amino acids is called peptide bond. So this peptide bond is amide bond, and it is a covalent bond. It is a strong bond. So during the formation of peptides or polypeptides or proteins, these amino acids, they share some of the electrons between them, especially nitrogen and oxygen, and they will lose one molecule of water. Suppose between this amino acid and this amino acid, when the formation of peptide bond, loss of the atoms in the form of water. So now we will not call them amino acid, we call them residues. The amino acids present in the peptides, proteins or polypeptides, these are called amino acid residues. So this peptide bond is nothing but it is amide bond, as I already mentioned. It is C double bond O NH. So this bond is peptide bond. Now this bond is trans configuration. When we see the structure of peptide bond, we will discuss all this. At this moment, you should know peptide bond is a strong covalent bond, and it is a bond between two amino acids, and the amino acids are called amino acid residues when after formation of peptide bond. So when two amino acids join together, there will be formation of peptide bond. So it is called dipeptide. So when three amino acids join together, we call tripeptide. So in tripeptide there are only two peptide bond. In dipeptide there is only one peptide bond. So like that, 4 tetrapeptide. So if many amino acids are there, we call polypeptide. Now we will see how this peptide bond is formed. So we know the structure of amino acids. So now peptide bond is formed between two amino acids. I will write the structure of peptide bond. So this is alpha carbon atoms. To the alpha carbon atom, there will be attachment of carboxyl group. So in physiological pH, they will not present as COOH. They will present as zwitter ion. So COO minus. For this alpha carbon atom, amino group on left hand side, because almost all amino acids are L amino acids, and it has got hydrogen and it has got R. So this is one amino acid. So I will name this as R1. Similarly, the peptide bond is formed between two amino acids. Here there are two amino acids. Literally peptide bond is formed between two amino acids. That means it is formed between carboxyl group attached to alpha carbon atom of one amino acid with amino group attached to alpha carbon atom of another amino acid. Or you can say like this, the peptide bond is formed between alpha carboxyl group of one amino acid with alpha amino group of another amino acid. So while doing so, there is lot of changes happening to this amino acid. So they will share some of the electrons, unpaired electrons between oxygen and nitrogen, and there will be formation of hydrogen bond. So there will be loss of atoms in the form of hydrogen and oxygen. So you can see this oxygen and the two hydrogen, they will removed as one molecule of water. So our peptide bond is formed between this carboxyl group and this amino group. So there will be formation of one water molecule. So this process is called dehydration, also called condensation. Imagine this COO can be written like this: C double bond O, CO minus. So this NH3 can be like this. Now there will be formation of one molecule of water. So remaining is C double bond O NH. So our peptide bond looks like this. Now we lost one oxygen, so we will write the carbon here and we will write the remaining C double bond O, will write like this. So this is with respect to one amino acid. Now here also there is remaining only N and H. This is our peptide bond. You can see this is actually one amino acid, this is another amino acid. So peptide bond is formed between this carboxyl group of one amino acid with this amino group of another amino acid. So now this is amide bond actually. C double bond O NH. So now see, this N is a peptide bond. So they say this bond is partial double bond because this bond is neither single bond nor double bond. That means in between single and double bonds. So the length of the single bond is 1.49 Armstrong. The length of the double bond is 1.27 Armstrong. The peptide bond here it is 1.39 Armstrong. So that means it is slightly more than double bond and slightly less than single bond. Because of this partial double bond, there is limited movement or twisting around this amide bond or peptide bond. But there are free rotation around alpha carbon atom. However, this depends upon this R group. If this R group is bulky, again the free rotation will be restricted. And please notice here this C double bond O and this NH, there is trans configuration here. Trans means opposite direction. Cis means same configuration. So C double bond O is in one side and NH is on another side. Almost all amino acids when forming peptide bond, they will form trans configuration. But there is an exception except proline. This is amino acid, it can form trans, sometimes it can be cis configuration. But majority of amino acids, when they form peptide bond, the C double bond O and NH is in trans configuration. That means opposite side. So this is good because they will keep away this R group away from one another. The conformation of the protein is for formation of second structure. This is very much important. The peptide bond is trans configuration. So because of this partial double bond, the limited movement, but there is free rotation around this alpha carbon atom, there will be conformation or spatial orientation around amide bond, also around alpha carbon atom. They will not be in a straight line like this. So the spatial conformation of this R group C double bond O NH will give a rotation to that peptide bond and other single bonds. I am going to explain that all peptide bond, all protein, we always start with N, that means amino terminal end, and end with carboxy terminal end. Because all amino acids, almost all amino acids in nature are L amino acids, amino group on left hand side. So this will become alpha carbon atom and this will become our carbonyl or carboxyl carbon. This is one amino acid. So now I will write another amino acid. This is alpha carbon atom. This is carbonyl or carboxyl group. Okay, this is another amino acid. So we know that there is this particular bond is partial double bond because it is slightly less than single bond, more than double bond. So there is a limited movement. But all these bonds are true single bond between N and alpha, C alpha. This is true single bond, and between C alpha and C, this is also true single bond. So because of this true single bond, so there is R group attached here. So this alpha carbon atom, one side amino group, another side is carboxyl group. This is R1. So the groups around alpha carbon atom, there will be free rotation around alpha carbon atom. So here R group may be this side. So this happens only because formation of this peptide bond. Even though there is a little rotation between these C and N because of partial double bond, but there is pre-rotation around this C alpha and C, N and C. Because of this true single bond between C alpha C and between N and alpha carbon C, there is a rotation. This is called angle of rotation. This angle of rotation is called Ramachandran angles. There is a famous Indian scientist during 1950 and during 1960. He did a lot of work and he plotted this angle. Name of this angle is called Ramachandran angles or angle of rotation. This is very important when we study the structural conformation of amino acids when they form peptide bond. Ramachandran angles. So there are two angles of rotation here between N and C. So this angle is called pi angle. And between C alpha and C there is again angle of rotation. So this angle is called psi angle. Pi angle and psi angle. So remember, the peptide bond is actually this one, C double bond O NH. It's a amide bond. There is a limited movement or twisting or rotation around this amide bond because this is a partially double bond, whereas there is true single bond between C alpha and C within the same amino acid, and also between the pre-rotation between alpha N and alpha carbon. Because of this rotation, so these R groups can occupy either place. This R2 can be here or it can be here. So there is a pre-rotation of R group between alpha carbon atom. However, whenever there is bulky amino acid, they have restricted movement. But this peptide bond, there is a limited movement because of partial double bonds. There is a pre-rotation between C alpha and C, between C alpha and N. Psi angle of rotation, and C alpha N, it is called pi angle of rotation. So this is Ramachandran angles. So when we plot, we can study the conformation of amino acid when they form peptide bond. So this is with respect to peptide bond. You should remember peptide bond is covalent. It is partially double bond. This is about peptide bond. Thanks for watching.